was no. I'm going to be referring to that talk a few times today, and it's available at the back. I think there are a couple of copies, and there's a sign-up sheet as well. And I'm not trying to self-promote, but if you're interested in that, there's a lot of information that I'm going to just basically highlight today, because this is sort of a sequel to that talk, and I didn't want to spend a lot of time on it. If you're interested, please see the book table. So, today my talk is titled The Mechanism of Adaptation. And if you remember last time, I sort of hinted at what it might be, but didn't really have an answer. Well, I'm going to answer that today. The subtitle is An Emerging Framework for Understanding the Engineered Optimization of Life. Those words are very carefully chosen, and you'll see why. So when we start talking about adaptation, a lot of people immediately think of evolution, and they ask the question, isn't evolution the mechanism of adaptation? So before we answer that question and get into the talk, I want to define some terms because there's some confusion about what adaptation is, what variation is, what evolution is. So let's look at those terms. We decided that I may have to turn and point at the back here to make this work. Maybe not. There we go. Okay, so let's clear up the terms. First of all, what is evolution? That is not evolution. There we go. That's exactly right. Okay, so the dictionary definition of evolution is as follows. Descent with modification from pre-existing species, cumulative inherited change in a population of organisms through time, leading to the appearance of new forms. The process by which new species or populations of living things develop from pre-existing forms through successive generations. So you'll notice it talks a lot about pre-existing species and new species. So the idea of evolution is what we would call modification. There we go, common descent. So it requires divergence via new forms. So the idea is that evolution creates new forms. It doesn't deal with the same forms. So you've probably seen a common, again, sorry about this, there we go, a common example of the concept of evolution, the evolutionary tree. The idea is that you started with one or a few species down here, and over time, new species are generated and they branch off from the original, so that up here you've got a lot of species that are not directly related, but they came from the same ancestor. That's the idea of common descent, and that's evolution. So in that, having said that, this is going to be a distraction, I'm sorry. So there we go. What is adaptation? So let's look at the definition of adaptation. It is adjustment to environmental conditions. An example would be modification of an organism or its parts that make it more fit for existence under the conditions of the environment. A heritable physical or behavioral trait that serves a specific function and improves an organism's fitness or survival. So it's about adjustment to environmental conditions. This is not the same as evolution. It involves existing forms that are adjusted, essentially improved fitness. So that's adaptation. There's another term we need to talk about, and that is variation. What is variation? Again, I'm going to go to the Merriam-Webster dictionary here. So variation is just divergence in the structural or functional characteristics of an organism from the species or population norm or average. So basically, it just is a deviation from the norm. So variation means an animal or an organism has a trait that goes off one way or the other from what the population is doing in general. So it's basically changing traits. It's just going deviation from the average. So what's important to know about these terms is the following. First of all, variation is not adaptation because variation is just change from the norm. It could be maladaptive, it could be adaptive, it could be helpful, it could be harmful. It's just change. It's just variation. It's not adaptation. Furthermore, adaptation is not evolution. Evolution requires common descent. So just because something is changing to fit its environment better doesn't mean it's evolving. It means it's adapting. And I've also heard the term microevolution to describe adaptation, but that's not accurate either because evolution, it's not a scale. It's whether or not something is turning into another organism. So there's no micro and macro. It's just evolution. Okay? All right, so obviously the most famous example of evolution, there are plenty, but is Darwinian evolution, right? So here's what Darwin had to say in describing evolution. It involves both variation and natural selection. So this is from the Origin of Species. There we go. This is from chapter 4. Here's what Darwin says. If useful variations do occur, can we doubt that individuals having any advantage, however slight, over others would have the best chance of surviving and of procreating their kind? On the other hand, we may feel sure that any variation in the least degree injurious or harmful would be rigidly destroyed. This preservation of favorable individual differences and variations and the destruction of those that are injurious I have called natural selection or the survival of the fittest. Now note, when most people think of evolution, they think of natural selection, but that's not what's going on here. It's variation and natural selection. And we just selected against the slide, apparently. There we go. Okay, so it's variation and natural selection. Both are very important in Darwinian evolution. But that presents a problem because Darwin recognized that he had no idea where variation came from, where it arose. Here's what he says in chapter 1 of the Origin of Species. He says, the laws governing inheritance are for the most part unknown. No one can say why the same peculiarity in different individuals there we go, of the same species or in different species is sometimes inherited and sometimes not so. Or why the child often reverts in certain characters to his grandfather or grandmother or a more remote ancestor, why a peculiarity is often transmitted from one sex to both sexes or to one sex alone, more commonly but not exclusively to the like sex. A lot of words to say, I don't know. So he doesn't know. He didn't know where that came from. But he had an idea. He had a theory or a hypothesis, and that was called Pangenesis. And so before he published The Origin of Species, he had this theory or hypothesis regarding the source of variation. And so he actually first started talking about it in 1836. And of course, Origin of Species was published in 1860. And he actually published Pangenesis in 1875 in detail. So what is his theory of pangenesis? It is as follows. So basically the idea is that every part of an organism generates these things called gemmules, and that those happen as a result of its interaction with the environment. And that happens at every single stage of development in the creature's life. That was Darwin's idea. These gemmules are then released from the cells, and they travel throughout the body, and they end up in the gametes, or the sexual reproductive cells, and then they're passed on by inheritance. That was his idea of pangenesis. You've probably never heard of pangenesis. The reason why you've probably never heard of it is because it was experimentally falsified by his own cousin just within a couple years after he presented it formally. So it's a false hypothesis. It failed. But obviously, evolution is still the primary or dominant theory about the origin of species right now. So what happened? Well, there we go. Evolution was widely accepted within a few years after origin of species. However, natural selection was not accepted as a driving mechanism between the 1880s and the 1930s, and here's why. Eberhard Denner, a German botanist in 1903, went so far as to say, we are now standing at the deathbed of Darwinism. And there's someone from MIT, Vernon Kellogg, who said, the fair truth is that the Darwinian selection theory, considered with regard to its claimed capacity to be an independently sufficient mechanical explanation of descent, stands today seriously discredited in the biological world. That was in 1907. Obviously, that's no longer the case. What happened? Well, what happened was, there you go. We just talked about it. Darwinian theory lacked a source of variation upon which natural selection could act. And as a result, there we go. As a result, that's why it was failing as a theory. However, it was rescued by the work of Fisher, Haldane, and Wright. And they were working between 1916 and 1932. They started the discipline called population genetics. And what they said was mutations are the source of variation. They increase the variability of the population. And they used their idea of mutations. They used statistical models with natural selection and mutation as the source of variation to come up with what's called the neo-Darwinian or the modern synthesis. But since then, obviously, that's almost 100 years ago. We've come a long ways, and there are now supposedly 5 evolutionary processes that are identified when you talk about evolution. What are they? Well, mutation is one, gene flow, non-random mating, otherwise known as sexual selection, genetic drift, natural selection. So these are the 5 supposed processes or mechanisms of evolution. So the question is, are these in fact mechanisms of evolution? And we're going to look at each one and find out. So let's look at mutation first. So this is a little graphic from Khan Academy that if you want to learn about evolution, you can go to Khan Academy and look at this. And this is the idea behind mutation that they illustrate. So basically, mutation is a change in genetic information. It's most often thought of as random. So in the little graphic here, you've got this population of beetles, and they're all the same genome type, and they produce the cells for reproduction, and a random mutation happens up here, and so one of them becomes black instead of white. That's the idea. The problem, though, is that the vast majority of mutations are either neutral or harmful. So the question is, do beneficial mutations exist? So you can't just make a mutation or a change. If it's not helpful to the organism, nothing's going to happen with it. It's just going to die out, right? And so are there beneficial mutations, and are they a mechanism of evolution? I'm going to ask you actually to hold that thought for 1 second, because I want to talk about these other first and come back to that. So let's talk about gene flow, another supposed mechanism. So in gene flow, the idea here is that you've got a migration of genes from one population to another. In the little graphic here, this little beetle saying "we" as it goes from one population to another. And so it involves you're taking existing genes and just introducing them to a new population. Of course, doing so does not actually create variation, it just redistributes it. And that's just as likely to be problematic as helpful. So what about there we go. What about non-random mating or sexual selection as a process? So that's the idea that you've got a preference for either similar or dissimilar mates. So and again, in the graphic up here, you've got a couple of black beetles that like each other and white ones, or you've got the opposite, right? The issue with this one is that it basically just involves existing genes that are grouped in an offsetting manner. So there's really nothing going on here in terms of variation. It's just redistribution, just like before. It doesn't change the relative frequency at all. And as a result, it's just as likely to be maladaptive as adaptive. So what about genetic drift? What's genetic drift, and is that a mechanism of evolution? Well, it's just random sampling of genes. So in this graphic here, you see you've got this population of black and white beetles, and basically due to chance events, maybe, who knows what the chance events might be, maybe the rock that they're all on falls off in the water and only 3 of them survive. So they're the only ones that reproduce, and you get all white as a result. So that's just random sampling of genes. There's nothing selective or purposeful about it. It basically just involves chance altering the size or the composition of the gene pool. Overall, it actually creates a reduction in variation because you're removing information from the gene pool. It has nothing to do with fitness to the environment. And again, it's just as likely to be maladaptive as adaptive. And it actually often reduces the viability of the population. So it's actually a harmful thing. And then natural selection. So what's natural selection? So natural selection, you've probably seen something like this or heard something like this. They often talk about predation as an example of natural selection. So in this example, you've got a dark-colored rock, you've got dark-colored beetles here and light-colored beetles. The idea is that they come along and the birds eat all of these light-colored ones because they can see them. And so when they reproduce, there's not as many white ones because the genes still exist for white, but most of the white ones are gone. So another way to look at it would be sort of conceptually as you've got your different variations, your random variation, you've got A, B, and C, and then the environment acts like a filter. So A and C are removed by the filter, they die, and B goes on to reproduce. That's the idea of natural selection. It's a filter. You've got random variation, and it filters. That's natural selection. The filter being, of course, death. So what should be clear from what we just talked about is that natural selection is really the only mechanism that they're proposing for evolution, because it requires a mechanism to act on the variation. And these others are just variation in that they redistribute or reduce the existing variation. They don't act on variation at all. So really the only mechanism we're talking about here is natural selection. So if we look at the ones that we looked at that are supposedly 5 evolutionary mechanisms: mutation, gene flow, non-random mating, genetic drift, and natural selection, what we discovered is that really mutation and natural selection are the only 2 that might have anything to do with evolution, if evolution in fact is happening. So let's look again at mutation, because natural selection is what's acting on the variation in the evolutionary model, but mutation again is what provides the variation in the evolutionary model. So the question is it the source of variation? It requires, for evolution, not just adaptive variation, but also novel. It's got to be new, otherwise species aren't going to diverge. And so ultimately you won't get common descent if you don't have new variation. So the question is in fact random mutation the source of novel or new variation? It is the source that's relied on in the theory. And it also has to, the theory of evolution has to have beneficial mutations. Not just mutations, but beneficial. They have to actually provide an advantage to the organism in order for natural selection to work. So the question is, are there sufficient beneficial mutations on which natural selection can act? And back in the 1930s, when Fisher and Haldane and Wright were doing the population dynamics and came up with the modern synthesis, neo-Darwinism, on the left is what they expected to be true of variations. So this is a magnitude of change, so not much change or not much change from the norm is in the middle and more change in the sides. They expected that there would be pretty much an even distribution between deleterious or harmful variations or mutations and those that are beneficial. This is what they expected. This is what they actually observed. Everything was harmful, nothing beneficial. That's what they actually observed. And this is the foundation for the modern synthesis of evolution. So the question is, it's been 100 years. What does that graph look like now? Well, here you go. This is from the 1930s, and this is at present. Now, it's a little hard to see, so I'm going to highlight it for you, but there supposedly are some beneficial mutations right down there. They're contested, but for the sake of argument, let's just say that those are, in fact, real beneficial mutations. The problem is, of course, the model was set up assuming basically 50-50, and now we've got very few that are beneficial. But there's a bigger problem. You may have heard of the term genetic loading. So this was a term that was coined by J.C. Sanford, and he was the inventor of the gene gun that's used in genetic engineering. And what he did is he expanded on a work by a guy named Kimura from 1979, and that work showed that most mutations are in fact nearly neutral. They're harmful, but they're nearly neutral. There's not much change from the norm there. And because of that, they're too small to be selected for or against by any kind of a mechanism like natural selection, for instance. So here's the graphic example of that. So this is the same chart as before. You see you've got the further it goes to the left is more harmful, further it goes to the right is more beneficial. They discovered that there's this no selection zone where it's too small to be selected, and that's right here in the middle. Now you notice something, that almost all the mutations are in that no selection zone, and the ones that aren't are harmful. And remember where the supposed beneficial ones are, they're right there. So what that means is that there are mutations that are happening, they're not being selected for or against, they're simply happening and staying in the genome, and they're almost all harmful. And that is what we call genetic loading. Basically, they rapidly accumulate these harmful mutations in the genome, and thus the genome actually rapidly breaks down, and that leads to extinction. So I hate to break it to you, but we're all going to extinction because there are harmful mutations building up in our genome. This is why, for instance, you don't have close relatives marry each other, because that reinforces the harmful mutations, right? So Sanford actually demonstrated this. He called it genetic entropy, and he demonstrated it in 2012 using H1N1 of the swine flu, and he showed that it rapidly went to extinction just from the fact that it was accumulating these harmful mutations. So there are, however, what a lot of people call mutations that do appear to be adaptive. So you see that an animal has adapted to an environment, and when you look in the gene, it seems to have a mutation that helps it adapt. But the problem with that is that these mutations, it's emerging that they're not in fact random at all. They appear to be repeatable, highly regulated, and tightly controlled by the organisms themselves. There's nothing random about them. So here's an example. This is from a chapter. This is the title. It says, "What is mutation? A chapter in the series, How Microbes Jeopardize the Modern Synthesis." Here's what it says. "The assumptions of purely chance mutations that occur constantly, gradually, and uniformly in genomes have underpinned biology for almost a century. But we argue that regulated mutagenesis mechanisms, so things causing mutations, greatly increase the probability that the useful mutation will occur at the right time and possibly in the right places. Assumptions about the gradual clock-like and environmentally blind nature of mutations are ready for retirement." So what this is saying is, or bringing the question is, are adaptive mutations really mutations at all? That's a fair question. They don't seem to be random at all. So hold that thought. We're going to come back to it later. You'll see why. So keep asking yourself that question as we go. Are adaptive mutations really mutations at all? Okay, so the end result of all this research that's happened and showing mutations are not what they appear to be, et cetera, is that because none of the supposed mechanisms except mutation are a source of variation for neo-Darwinism, for evolutionary theory, and because random mutation does not provide selectable variation, it only results in genetic loading. Without novel selectable variation, natural selection cannot result in divergence in common descent. So evolution cannot happen. Because of that, just like Darwinism before it, neo-Darwinism has no viable mechanism and therefore is an invalid theory. And this is being recognized by evolutionary biologists all over the place. So here's a few examples of papers and books that have come out recently. Darwin retried an appeal to reason, what Darwin got wrong. Darwinian evolution is in the light of genomics. The types, a persistent structuralist challenge to Darwinian pan-selectionism. Dogma and doubt. Does evolutionary theory need rethink? And an expose of the evolution industry. Let me give you a few quotes from that last one. So Susan Mazur says, "Scientists agree that natural selection can occur, but the scientific community also knows that natural selection has little to do with long-term changes in populations." Antonio Lima de Faria says, "Darwinism and the neo-Darwinian synthesis, last dusted off 70 years ago, actually hinder discovery of the mechanism of evolution." Now, they haven't gotten rid of evolution, they just recognize it has no mechanism. And Jerry Fodor is even more blunt. He says, basically, I don't think anyone knows how evolution works. That's a problem. So I'm going to go out on a limb here and say that evolution is not the mechanism of adaptation. They have no mechanism in the theory. So it's not a good theory. We're going to dismiss that as the mechanism of adaptation. But then the question becomes, remember, evolution requires variation and natural selection. So we've talked about mutation is not providing novel variation. So the question is, what about natural selection? Is natural selection the mechanism of adaptation? And why do I ask that question? Because it could have a different role. In evolution, because it's descent with modification, requiring divergence and common descent, it requires the novel variation plus natural selection. But what if, when we're just adapting to environmental conditions, we don't actually need novel variation, we just use existing variation plus natural selection? Would that work? Because that would be a mechanism for adaptation. And that's actually the role that Susan Mazur just said. You know, everyone, all the scientists know about natural selection, but it has nothing to do with evolution, basically, is what she said. And actually, most creationist models of adaptation also assume this kind of role for natural selection. So if you go to AIG or Creation Ministries International, they're going to tell you natural selection is real, and that's what God uses for adapting creatures. That's what they're going to say. And typically, when they do that, they say, well, natural selection is real. It's obvious. Just go look at predation. So let's look at predation. I'm going to give you a couple of classic examples of predation that supposedly is natural selection. The first is a gazelle and the cheetah, right? So you've heard this one. You've got a bunch of gazelles, And originally the gazelles weren't that fast. You had the cheetah, and the cheetah would go and eat gazelles, and the only ones that survived were the ones that could outrun the cheetah. And so over time, the gazelles got faster and faster and faster, and then so did the cheetah, right? Because the cheetahs that weren't fast enough couldn't catch the gazelles, so they died too. And so over time, you get a fast gazelles and fast cheetahs, right? The problem is that's actually not how it works. The problem is that predators typically prey on the young. So the traits of being really fast aren't fully developed yet, right? And as a result, actually what happens is the predators are opportunistic, right? The prey are in the wrong place at the wrong time. It doesn't really matter how fast they are, honestly. That's not a huge consideration. So it's maybe some selection going on, but it's inefficient at best. So what about the peppered moths? This is another great example that's often trotted out about natural selection, right? So you've seen peppered moths before. You've seen these pictures. Here's a peppered moth on a tree. Actually, there's two peppered moths in this tree, believe it or not. There's the black one, and right down here is the white one. I don't know if you can see him or not, but he's obviously very well camouflaged. Well, of course, if you switch out the tree trunk, now the black one is very hard to see, but the white one's obvious. And the idea is the Industrial Revolution, you got blackened tree trunks, so the frequency of the population changed because birds were now seeing the white ones and they were picking them off and eating them, so the population got mostly black, and then after the Industrial Revolution, the tree trunks got cleaned up and turned back to white, and now the population is mostly white again, right? You've heard that, right? There's a problem with that. The caterpillar is also actively camouflaged. It's not just the moths. So here's a picture of the caterpillars. This is the caterpillar for the dark one. See him? Very well camouflaged against those sticks on the tree. Here's a caterpillar for the white-colored one. And again, in the greener twigs, he's camouflaged extremely well. The caterpillars know this. And so the green ones hang out with the green twigs. And the black ones hang out with the dark twigs. They're smart that way. They don't just sit there and wait to be eaten. And as it turns out, the moths are the same way, right? I mean, as expected. They know they're camouflaged, and they use the camouflage backgrounds, and they even rest beneath leaves. The pictures of them sitting in the tree trunks is a very unusual circumstance, and actually some of those pictures were faked. They were just pinned to the tree to try to make a point, right? But that's not how they actually work. So again, just like the gazelles, the caterpillars and moths that are eaten are in the wrong place at the wrong time. They're not being selected based on their camouflage, they're being selected based on whether or not they're in the right place, because the predators are opportunistic. So both of these classic examples are better characterized as examples of genetic drift, random sampling. So essentially what's happening is predation is happening regardless of their genetic traits. It happens to do with their behavior and the behavior of the predators. That's what it boils down to. And we can go into a lot of different examples about what seems to be natural selection really isn't. But instead of doing that, because we're actually going to be focusing on the mechanism of adaptation, I'm just going to reference back to my presentation from 5 years ago. Does natural selection drive the origin of the species? So basically, I went through an entire presentation, an hour and a half, talking about natural selection and giving lots of examples and showing how natural selection doesn't seem to be what's actually happening. So first of all, I pointed out that natural selection assumes that adaptation results from slow filtering. And where does the idea come from, slow filtering? Well, it comes from Darwin himself. Back in chapter 15 of The Origin of Species, he says, as natural selection acts solely by accumulating slight, successive, favorable variations, it can produce no greater sudden modifications. It can act only by short and slow steps. That comes straight from Darwin. And we looked at that in that presentation, and what we discovered is that observed adaptation is not like that. It's fast. And in addition to that, it's repeatable, it's reversible, and it's precisely controlled. It's not random. So the conclusion was that differential survival, which is natural selection, is what they mean. Some things survive, some things don't. There's differential survival. It's not really a filter. It's too slow, it's too broad scale, it's too unpredictable, and it just can't efficiently and effectively sort variations. And again, I'm not going to dwell on this. If you want more detail about that, the presentation from 5 years ago is back at the book table. So the bottom line here, again, I'm going to go on a limb based on what I said 5 years ago and say natural selection is not the mechanism of adaptation. So that begs the question. If there's no selection, then what's going on? Well, let's go back to this conceptual diagram that I brought up earlier. So natural selection is seen as random variation plus differential survival. And we just said that natural selection is not what's going on, right? So we're going to eliminate that. No natural selection. So now we're left with random variation. But we just talked about how variation doesn't appear to be random if it's adaptive. So we're going to eliminate that. So what are we left with? Variation, right? So the bottom line to that is, when we ask the question, what is the mechanism of adaptation, what we're really saying is, if I can get to it, there we go. Oh, what we're really saying is the presentation's over, apparently. There we go. We're saying, what is the mechanism of variation? That's really the question. Remember, that's the same thing Darwin struggled with. He didn't know where variation came from, and neither do the neo-Darwinists. So let's see if we can answer this question. We're going to start with a classic example of variation, beak variation in Darwin's finches. So this is an example of the different finch varieties, some of them, on the Galapagos Islands that Darwin saw. And you can see, obviously, the biggest difference is the beak size, right? And what has been discovered in the course of study of these finches is that there is a correlation between the beak variation and their habitat and diet. And so this is just a chart that shows you that. You've got your vegetarian finches over here, insectivorous finches, et cetera. You go around these different kinds of finches, and they can be organized by what they eat: seed eaters, cactus eaters, insect eaters, fruit eaters, and by their habitat in the tree, on the ground, et cetera. So there's a lot of correlation there. And you've probably heard all of that before. What most of you probably haven't heard is there's another correlation, too, that's been interesting, that's been observed, and that is there's a correlation of beak depth and precipitation. So what's beak depth? That's the height from top to bottom of the beak. And what's been interesting is that studies have been done since back in the mid-70s, and when there's a dry year, the population, their beak depth is greater. And then it kind of goes back to normal. Another dry year, it peaks again, same thing. And then a wet year, it goes the opposite direction. So the beak depth varies with the precipitation in their climate. Very closely. There's a very close match. So that begs the question, what's going on here? And does that hold the key to variation? The beak variation is happening, and it seems to be closely tracking with the environment, and precipitation specifically. So here's what's interesting about that. That variation is not random. It's precisely defined and controlled. Why do I say that? Because someone went and studied it. And what they discovered was that the beak variation is not random and not sort of chaotic or haphazard. In fact, it's very tightly controlled, such that you can actually do a study and take all the different beak shapes and actually reduce them all to a conic section, a parabola. So they all fit on a parabola. They don't deviate from the parabola. So here's what the study authors had to say about it. They said, to further quantify the diversity of shapes, we fit polynomial functions to the beak profiles and search for the beak shape with the simplest functional form. This turns out to be the Geospiza in the Darwin's finches, which are fit well to within the error of our methods for recording shapes by a parabola. There's the formula for it. Y equals x squared plus bx. The shear collapse of all other songbird beaks, not just Darwin's finches, onto this shape implies that all beak profiles are well fit by an equation of the form 0 equals ax squared plus bxy plus cy squared plus dx plus ey. So what's that saying? What that's saying is there's not random variation going on. They're all parabolic sections. They're geometrically, mathematically, functionally defined. That's really interesting. In addition to that, they go on to say, the present study significantly limits the potential set of parameters that can be optimized, since the intrinsic developmental pathways place strong constraints on the set of beak shapes that are actually produced. An optimization, or any optimization, can occur only within the subspace of shapes that is readily available to the existing beak developmental pathway. That's a lot of words to say that this happens in embryo, in development. The variation that occurs in these beaks occurs during development in embryo, and it's precisely controlled with mathematical precision. So basically, the appropriate beak morphology to fit the environmental conditions is chosen and precisely controlled by the mechanism itself, the organism itself. And it happens during embryonic development, has nothing to do with reproduction. Of course, natural selection has everything to do with reproduction. Nothing to do with it. It happens in development in the embryo. This is best characterized as engineered optimization, and you'll see why I say that in a minute. So this leads to a couple hypotheses about the mechanism of adaptation. The first is that adapted variation is an engineered response to environmental change. The second is that adaptation is not for the purpose of survival, but for optimization, and survival is just a side benefit. So what do I mean by optimization? Well, here's the definition for that. Optimization is an act, process, or methodology of making something, such as a design, system, or decision, as fully perfect, functional, or effective as possible. Specifically, the mathematical procedures, such as finding the maximum of a function, involved in this. Optimization is a mathematically precise design feature, okay? And keep in mind this finding the maximum of a function. We'll come back to that later as well. Okay, so I'm going to propose a framework to help us understand what's going on with adaptation. And I'm calling it the Ttam Framework. And this is describing what's going on that's engineered optimization in living things. So what do TTAM stand for? Well, T is for tracking of environmental conditions. I'm going to assert that organisms track environmental conditions and that adaptation is thus responsive, not passive. The second T stands for targeting of solutions to environmental problems that these organisms encounter. And I'm going to say there that adaptation is targeted, not trial and error. It's purposeful, it's targeted. A stands for assembly of traits. Traits, of course, being the features of the organism that happen to be adaptive to the environment. And I'm going to say there that adaptation occurs during development, not reproduction. And the M stands for modification of those traits. Adaptation is adjustable, not static. Okay? So let's talk about this first one. The assertion is that adaptation is responsive, not passive. Why do I say that? Well, I wish I had a lot more time to go into detail on this in particular, but let's just take my word for it. You can do your research if you want as well. I'll give you a couple of examples, but organisms are equipped with an incredible array of sensors, and they're tuned to specific environmental variables. This is an emerging field of study. If you want some more information on this, you can look at there's a series of Acts and Facts articles by Dr. Randy Galuza called Engineered Adaptability. You can go look at those, and there's lots of other places to find it. But let me just give you a couple of examples. These include organisms that can detect the polarization of light and magnetic field direction and strength. They use this for navigation. You're familiar probably with birds and butterflies. The monarch butterflies navigate all the way from Canada to Mexico with the magnetic, by sensing the Earth's magnetic field. It's a lot more complicated than that. There's actually multiple systems. They detect magnetic strength. They detect magnetic direction. They make a map of magnetic field, local magnetic field changes, and actually in salmon, they make a map as they go out to the ocean, and then they come back, they follow that map till they can get close enough to smell their home waters, and then they actually pass that map on in inheritance to their offspring. The offspring have never been to the ocean, but they know how to get there because they have a map that their parents made. It's pretty amazing. In addition to that, there's lots of sensors for things like humidity, temperature, salinity, conductivity, fluid flow, etc. And these are often used for habitat suitability, for determining habitat suitability. And of course, organisms sense things like pheromones and chemicals to avoid predators, find mates, etc. So there's a lot of different sensors going on. And again, I wish I had a lot of time to talk about that, but we're actually moving on because of the mechanism of adaptation is our goal. But suffice it to say that sensors detect very specific environmental conditions, and they're predetermined as stimuli for those sensors. There's nothing chance about it. These sensors then trigger a biochemical response cascade, which results in something happening in the organism once that stimulus threshold has met. So it's not just the fact that it's a stimulus, there's a threshold. It has to meet a certain threshold, and once that's met, it triggers a sensor and biochemical things happen in the organism. Okay, so again, I wish you could spend more time on that, but continuing on about tracking, adaptation, again, is responsive, not passive. Organisms actually have the characteristics of a tracking system. So think, for instance, like an anti-missile battery. Those systems have sensors which detect based on pre-specified conditions. If you see a plume from a missile, it detects the heat, for instance. And in addition to that, they have algorithms which determine, okay, if I detect this, what do I do? And then they also have, that's the opposite of no input. They get lots of inputs. They also have outputs, so they adjust an internal state or an external behavior based on them sensing and having logic about what to do about it. So that's a tracking system. Well, of course, remember Darwin's finches. I have to say this is actually about the most technical difficulties I think I've ever had speaking, so sorry about that. It's very distracting, but we'll try to get through it. So I know those guys are doing their best to make it work. It's okay. You do not want to see me dance, trust me. My children tell me that all the time. Don't dance, Dad. I don't know how to do that. Okay, should I just take it back to where it was? All right, let me see if I can move it. There we go. Okay. So remember Darwin's finches we just talked about, right? They closely correlate the beak size with what's happening in the environment. They're tracking the environment. They're sensing what's going on. They've got logic that tells them what to do once they figure out what's going on, and then they have a response. They change their beak size. So adaptive variation is not only tracking, but also targeting. What I just talked about, they're basically targeting the beak size for the appropriate conditions. So adaptive variation is rapid, predictable, repeatable, reversible, and precisely controlled. What do I mean by that? Well, let's give some examples. All of these examples I'm about to give are rapid. They happen very quickly. It's sometimes as little as one generation. We're talking like, a couple of years. And it's pretty dramatic, the changes. But let's talk about the predictability of adaptation. So let me give you just a couple of examples of that. This is a Gymnodactylus amarali, a gecko, okay? And there's a study done on gecko when there were 7 new islands created and something strange happened to the gecko populations. So basically there's a hydroelectric project that flooded an area and made 7 islands. So here's a map of what it looked like. This is in South America, so you've got these islands that show up here. There's a dam here. So it flooded this area. There was 1 population of geckos in this area, and now there are 7 isolated, actually 8, because these are all populated, isolated from each other, and isolated from the mainland. So there's now 8 populations of geckos from an original first population. Something else then happened, and that is there was a species of lizards on these 7 different islands that was preying on larger termites that the geckos didn't eat because they were too large. And that species went extinct. Well, predictably, what do you think happened? The geckos on all 7 islands developed larger heads to prey on that now available food resource. It happened in 15 years. All 7 populations, unrelated and isolated from each other, did exactly the same thing. It was predictable. And there's no overlap. This is the mainland head size, and this is the island head size. There's no overlap here. It's a very, very clear response to what happened. It happened very quickly, 15 years. Here's another example. This is an example of adaptive tail lengthening in forest deer mice. So there's basically 2 main habitats for deer mice. There's prairie and forest. Here's a map that's showing you that. You've got prairie in the middle, you've got forest is green, you've got a western population, or a western habitat, and an eastern habitat of forest, right? 2 totally separate populations, east and west, not related. Turns out that there are longer tails and caudal vertebrae in the forest variant compared to the prairie variant. So here example is the prairie variant, this tail ends right there, and this is the forest variant, longer tail, longer vertebrae in the tail, and you see there's no overlap. This is prairie, this is forest. Same thing in the east, shorter tail in the prairie, longer tail in the forest. Well, as it turns out, this is exactly what should be expected because the longer tails are advantageous in an arboreal or forest habitat. And the same adaptations occurred in the unrelated populations on east and west, very predictable. So that's just a couple of examples of predictable adaptation that happens in response to the environmental conditions. But I also want to talk about something that's even more interesting, and that is it appears that organisms have what you would call solution sets to problems they encounter. So these solution sets are predictable, they're repeatable, and they're often reversible, and they're used over and over and over again as the same organism adapts to changing and novel environments. So let me give you some examples. There you go. So we're talking about locusts, dung beetles, butterflies. There's a moth, E. coli, common bacteria, E. coli, Hawaiian stick spiders, fig nematodes, rove beetles, Omicus, which is rainbow trout and steelhead, three-spined stickleback, which is another fish, Mexican blind cavefish, blind centipedes, and salamanders. And I'm actually going to go through all of those. So let's get started. So this is the locust that has two forms, the solitarius and the gregarious. What does that mean? Well, that means that these are lonely and these are not. So what it really means is that this is the form, this color shows up when there's not a high population density. When the population density goes up, this form shows up. So these two forms diverge in response to population density. Okay, another example, horned and hornless forms in dung beetles. It's a little hard to see on the screen here, but this has a giant horn sticking out of it, and this one does not. So two populations, they either become horned or non-horned, and they diverge in response to the size they have as larval body size. And here's another example. You've got winged and wingless forms in aphids. Again, these are predictable and repeatable solution sets to the same problems. Here's your wingless form, and there's your winged form over there. And these two forms diverge in response to actually a number of things: population density, plant quality, temperature, and photoperiod. Let me give you another example. Now, you might think you're looking at 6 different butterflies here. You're actually looking at 3 different species. Each species has 2 forms. This is one species, that's another species, that's another species. They have change in color and wing pattern, and that is dependent on or triggered by change in seasons. So if they're born in the fall, they've got one. If they're born in the spring, they've got another. Same genes, same species, 2 different solution sets to the environment they find themselves in. And here's one of my favorite ones. This is the moth. This is the Nemouria Arizonaria moth, so obviously it lives in Arizona. And you can see that it's pretty well camouflaged in the leaf it's on, right? That's the moth. The interesting thing isn't the moth, though. The interesting thing is the caterpillars that diverge into 2 separate forms based on season and diet. So these moths go and they lay eggs in oak trees, and they either lay them in the fall or the spring. And here's what happens. There are 2 caterpillars in this picture, one on the left and one on the right. Here are the oak blossoms on the oak tree. Here's the caterpillar. So, and here on the right, you've got twigs of oak, and here's the caterpillar. Same genes, 2 different seasons. What has happened here, these caterpillars actually were born, when they hatched, they looked the same. They started eating their food source, and they became, in this case, it started eating the blossoms and started looking like the blossoms. In this case, which is even more amazing, it eats the leaves and then starts looking like the twigs that the leaves are attached to. So this is probably the most incredible example I know of you are what you eat. Pretty amazing adaptation. Okay, so let's move on and look at some more here, if I can get it to work. There we go. So E. coli, I mentioned E. coli earlier, which of course is a bacteria you want to avoid if possible. It'll make you very sick. But E. coli is great because you can do a lot of experiments on it. Reproduces very quickly, it's easy to work with. So some experiments were done in which they did 3 separate populations of E. coli in 3 different test tubes, completely isolated environments, no interaction whatsoever, same source population, 3 test tubes. And here's what they did. They fed these E. coli 2 different kinds of food, an easy-to-digest glucose and a hard-to-digest acetate. Now, the E. coli can switch in between these 2 different foods. But what they discovered was, in all 3 populations, in all 3 test tubes, when they did this, gave the 2 different kinds of food, what did they end up with? They ended up with 2 specialized forms in each test tube, one specialized for glucose and one specialized for acetate. Happened repeatedly, all 3 populations. But even more amazing, what they discovered is, when one of those food types got exhausted, guess what? They adapted the ability to switch between the 2 food types again. Interesting. So here's what the study authors had to say about that. It said the population is diversified into 2 existing ecotypes representing different physiological adaptations. We found that similar but independently evolved phenotypes often shared mutations in the same gene, and in 4 cases shared identical mutations at the same nucleotide position. Interesting. Here's what Nature News had to say about the same study. It said, in the new study, the authors went back to the frozen samples from 3 of their test tubes and sequenced 17 gene samples from various stages of the experiment. The DNA showed that in some cases, identical mutations appeared independently in all 3 test tubes, despite the random nature of mutations. The same changes in the environment favored the same genetic solutions. I'm sorry, but that's kind of gobbledygook. You're saying that identical mutations happen despite the fact that they're random, which makes no sense. And then you're saying the environment favored these supposedly random mutations that happened again and again, identically. So the reason for the gobbledygook is because they don't know what's going on. This is not what was expected. This is not the way E. coli is supposed to behave. It's supposed to be random mutations and natural selection, but that is not what's happening. They're struggling, trying to figure out how to describe this. So Nature News goes on to say this. Dobelli and Heron also found that some mutations occurred only in a specific order. After one group had become specialized for glucose and the other for acetate, both groups evolved to switch better between meal types. The last mutation would not have been useful until after the emergence of the first, which helped exhaust food supplies faster. So they're saying that not only are they identical, but somehow random, and somehow favor the environment, but also they occur in a specific order every time, and just repeated again and again and again. Something going on here that's not random. Here's another example of predictable, repeatable solution sets being employed by organisms. This is a Hawaiian stick spider, and these inhabit several islands in Hawaii, from Kauai, Oahu, Molokai, Maui, and the Big Island of Hawaii. And they have come in 3 different camouflage variations: the shiny gold, which you see here, dark, and matte white. So here are some examples of those spiders from different islands. So the top row is from Kauai, this is from Oahu, these are from Maui, this one's from Molokai, and then these are from the Big Island. So you'll notice here they got a little inset to show you the kind of camouflage. There's only the 2 matte white here, and that's a close-up of the camouflage. They have the same exact camouflage, but you see their body is different. See that? Different body, same camouflage. These are all the dark ones. Again, an inset of what the camouflage looks like. So look at this one, all curly body here. Totally different than these body types. And same thing here, very strange body types compared to the others, but all have the same shiny gold camouflage. So even though they're implying completely different body types, there's 3 types of camouflage, and they have one of the 3. But here's what's more interesting than that even. That is that the spiders appear to have colonized the Hawaiian Islands in sequence. And if you're familiar with how geologists think Hawaiian Islands formed, there's a plate that apparently moved over a hotspot, and Kauai was formed first, then Oahu, Molokai, et cetera. Now, of course, we can argue about how long that took, but that's a side discussion. In any case, so it appears to be evidence that the spiders colonized starting at Kauai and moving down to the Big Island of Hawaii. But here's what's even more interesting. The same 3 colors are produced in all of those islands, regardless of which color colonized the island. So if a dark spider went to the next island, instead of having all dark spiders, you have dark, shiny gold, and matte. And if a shiny gold one went to the next island, instead of having shiny gold, you have dark, shiny gold, and matte. So it happened again and again. And so here's what the Science Daily has to say about this study. It says, we don't usually expect evolution to be predictable, but Hawaiian stick spiders of the Aramines genus have repeatedly evolved the same distinctive forms, known as ecomorphs, on different islands. This repetition suggests that the Arizonaria spiders have some sort of pre-programmed switch in their DNA that can quickly be turned on to allow them to evolve rapidly into these successful forms. But how that process might work is still unclear. Okay, they're saying that this is not an accident, this is not random, this is not natural selection, this is a pre-programmed switch in their DNA. Interesting. But it gets better. So these are just some drawings of 3 different species of fig nematodes, and these occur all the way from Australia to Vietnam. And then on the right here are actually some pictures of those various forms. So you see there's 5 different forms from one species, and the same 5 forms, essentially, they're analogs from another species, very widely separated. Same thing down here. This one's kind of an abnormality, but it does correspond with this other form. Say here you can see the same thing. These look similar. So here's how this happens. The fig nematodes, they colonize figs after they arrive on pollinating wasps. And in the first generation, when they get to the fig, they all look the same. They have a tube-like mouth, and they feed on microbes. But The second generation, depending on where they're located in the fig and what they eat, turn into one of these 5 forms. And the genetics are exactly the same. These are genetically identical, but they look totally different. This happens again and again and again every time these nematodes colonize the figs. Same 5 forms happens again and again and again. I think we may have just lost the picture again. Yes, we did. Right before I was going to give you my best example yet. Okay, here we go. So, these are rove beetles. Now, rove beetles are parasitic on ants. And they do something very interesting. They anatomically mimic the ants in order to be accepted into the colony, where they eat the larvae and the food resources. And they also mimic ant behavior and pheromones in order to be accepted. On the left is what a rove beetle looks like when it's just by itself, not in an ant colony. On the right is an example of a rove beetle. This is not an ant. This is the same rove beetle, same type of organism that has changed its anatomy to look like an ant. And down below, you might think that's 2 ants. That's not. That's an ant and a rove beetle. Can you pick out which one's which? Well, let me help you. There's the rove beetle. So they're amazing at mimicking the anatomy of ants in order to prey on them. And these kind of changes have happened at least a dozen separate times. And they're very specific to the target ant, such that they identified in this study 9 different variations of that same rove beetle, depending on which ants they are preying on. Those are all the same thing, all rove beetles. Look totally different. So here's what the study authors had to say about this one. So in reconstructing their evolutionary history, again, still on the idea of evolution, but actually this is adaptation, not evolution. We uncovered evidence of conspicuous repeated evolution over deep time that runs counter to the notion of evolutionary contingency and represents a new paradigm for understanding the origins of interspecies relationships. Science Daily, commenting on the same study, says, this discovery provides evidence that evolution has the capacity to repeat itself in an astonishingly predictable way. The tape of life has been extremely predictable whenever rove beetles and army ants have come together. It begs the question, why has evolution followed this path so many times? Because it's not evolution. Because it's a predictable, repeatable response to environmental conditions. Another example of predictable, repeatable, but also reversible solution sets. On the left are rainbow trout. On the right are steelhead. It turns out they're actually the same species. And the steelhead, of course, go to the ocean and come back, and they become smolts. They go through a smoltification process where they change color, they change size, they get the ability to be able to filter salt out of the water so they can survive in the ocean, and then they leave. The resident trout don't do any of that stuff, they just hang out. But what's interesting is, before that happens, they're virtually indistinguishable as juveniles. And then, still trying to figure out exactly why, but some combination of factors like temperature, flow, growth rate, and distance to the ocean causes these anadromous ones to switch to anadromous and go to the ocean and come back. And the interesting thing about this is that you can have anadromous parents, so steelhead parents, that give rise to rainbow trout offspring. Or the reverse, rainbow trout parents that give rise to steelhead offspring, or any combination thereof. And this occurs even when they're isolated for 70 years, for instance. In an experiment, they stocked a lake with steelhead, had a barrier fall, so any of the fish that left to go to the ocean couldn't come back. Over time, those fish all turned into rainbow trout, resident trout, and that was the case for 70 years. After 70 years, they allowed those rainbow trout to go to the ocean, and guess what? In one generation, they had steelhead. So it's reversible. Here's another example, 3-spined stickleback. This is a fish that has both marine and freshwater variants. The marine form is larger, darker color, has a lot of armored plates, and a pelvis with 2 long spines. The freshwater version is smaller in size, is lighter in color, it has few or no armor plates, and it has a reduced or remnant pelvis which doesn't have any spines. So here's just a drawing of what that might look like. You got the marine up top, the red are all just they're colored for emphasis, are all the armor plates, and here's the freshwater version down there with very few armor plates on it. What's interesting about this is there we go, that the loss of the armor plate and the spike feathers has happened multiple times as the marine forms colonized freshwater habitat over and over again. And in addition to that, the marine and freshwater variants still interbreed on coastal rivers, and they still produce both marine and freshwater offspring. It's repeatable and reversible. Here's probably a famous example. You've probably heard of this one, blind cavefish, the Mexican tetras. Here's the blind one up here. You can see it doesn't have any eyes. I don't know if you can see that from there, but it doesn't have any eyes, and it's very light in color. And here's the sighted version of that fish that lives in the rivers nearby. So the blind cavefish descended from those river populations. What's been discovered is that the degrading or absence of the eyes is correlated with a specific protein called the HSP90, it's heat shock protein, which is a protein that the fish produce as a stress response. And that happens during development, while they're still in the egg. What's interesting is that both the adult fish and the embryos exhibit this response in when they detect lower conductivity of water, which caves have water with lower conductivity that's been discovered. And in addition to that, the river fish have the same exact response to water with lower conductivity. So that it's also the case that blind cavefish can produce sighted offspring in as little as 1 generation. Now, blind cavefish have often been used as an example of natural selection with loss of information, or they've lost information to do eyes, et cetera. That's actually not what's happened. What happened is they all have the information for eyes, but the blind cavefish sense their environment of lower conductivity, respond with stress. That stress causes inhibition of that stress protein, and that degrades their eyes, so they're born without eyes. It's predictable, it's repeatable, and it's reversible, and it's a response to their environmental conditions. But what's interesting about that is blind cavefish aren't the only animals that do this. So in caves, you've got your blind cavefish, of course, over here, but you also have, as an example, a blind centipede. You note the lack of color compared to most multicolored centipedes that you see outside. And also, here's another example, there's lots of them, but here's a Texas blind salamander. Again, salamanders are generally brightly colored. This one is not, and it's also blind. So what we have going on here actually is, there we go, they have a loss of eyes, skin pigmentation, and studies have also showed a loss of circadian rhythmicity. It's a handful of a word to say. And in exchange for that, they have an enhancement of wakefulness, taste, smell, and mechanosensation. In other words, they can sense vibrations and such. So what's happening here is you've got a similar condition, a cave, lower conductivity water, and similar internal programming across these widely varied creatures, fish, centipedes, and salamanders, and you get a similar response. What's happening is here, you've got engineered optimization going on in response to environment. That's what's happening. All right, so we've gone through a lot of species that are basically the same species that you can see how they have solution sets they employ to respond to their environment. But what about species in general? Are species also solution sets? And if so, does that mean speciation is reversible? Because we just talked about predictable, repeatable, reversible. So the answer to that is yes and no. What do I mean by that? Well, closely related species can and do merge. In fact, there's a great example of Darwin's finches. There was one island that had 3 different species of Darwin finches, and as it turns out, those 3 species are merging back into one. So that can happen. But there are also examples of species that have solution sets that don't seem to be overlapping, and so they have a hard time merging back together. What's an example of that? I'll give you a few examples. This is the genus Panthera. Of course, you got your lion, your tiger, your jaguar, your leopard, and your snow leopard. These are different solution sets, or so it would appear, to environmental issues that these cats encountered. But what happens if you try to mate these together? You get a hybrid, and there's a couple of examples. If you take a male lion and a female tiger and mate them together, you get something called a liger. And if you do a male tiger and a female lion, you get a tigon. And they're kind of strange looking creatures. So first of all, a liger is the largest cat by far. of any cat out there. It's gigantic. So here, example, is a liger next to a tiger. It's a very big cat. But you notice it's kind of weird. It has sort of like a lion head and sort of tiger stripes, and it's kind of strange. Here's a tigon, and again, it has a tiger's head, I think, and some sort of stripes that are sort of a mashup between, if you look at a lion real close, they have kind of a speckled pattern to them. So this seems to be a mashup between a tiger stripe and a lion speckle. It's kind of strange. But those aren't the only strange hybrids. These are zonkies. This is a zebra and a donkey. So you get a donkey with zebra stripes. What's even stranger is here's a donkey, there's its mom. Doesn't look much like mom other than the stripes. You also get zorses, which are even more bizarre. So here's a zorse, a donkey and a horse. He's got the zebra stripes. A zebra and a horse. And here's one that looks like it's kind of a zebra on the head and sort of the rump and a horse in between. It's kind of a strange mashup, right? But there's more examples. This is something called a beefalo on the right. So here's a buffalo. This is a picture I took of a buffalo in Yellowstone last year. And here's a beefalo. Now you notice that a buffalo has a very large shoulder and fuzzy fur on the shoulder compared to its head. Same thing here. Smaller head compared to the large fuzzy shoulder. Here's another example of a beefalo, because they don't all look the same. Again, a smaller head, large shoulder, fuzzy face, but look at this, the mashup between a cow's color and a buffalo's color. It's kind of weird. Okay, so these are a mashup. They're sort of like a Franken-car, right? So you've got here you got your off-road vehicle, your SUV, and your sports car all mashed up together. Or what's more my favorite is a Lamborghini tractor. So I would love to have one of those. That just looks cool to drive. So these are weird mashups, right? So what's going on with these? Well, actually, there is a pretty straightforward explanation for this, which is found in the Mechanism of Adaptation. But before we get to that, we're going to take a break, especially since I'm a little longer because we've had technical problems. After the break, we're going to finish talking about the Ttam Framework, and then finally, we're going to identify the mechanism of adaptation. So.
Yes, this is amazing. You used the word amazing. It is amazing. Thank you very much, Phil.
It's going to get better.
Take a break until 10:30, and we'll get back in here at 10:30. And beverages are allowed with lids on them, so see you back at 10:30. Thank you.
Address the elephant in the room again, which is the technical difficulties we're having. These guys have a new system they're trying to work with, and they're having some troubles. So we will do our best to get past those and not be a distraction from the talk. It probably will take a few minutes longer than I had planned because obviously, as you can see, my slides don't change when I ask them to. So it's going to take a little bit more time. So I'll give it about 30 more seconds, and then we'll go ahead and get started. What do you guys think so far? Yeah, pretty amazing. Yeah, it's pretty amazing stuff. But I know the applause is not for me, it's for what's going on that I'm talking about, the content. It's incredible the way God has designed things. Me too? Well, thank you. You're very kind. Thank you. It's incredible the way God has designed things to adapt to their environment. I mean, it's amazing. So far, I've talked about lots of examples, but hopefully I'm about to blow your mind with how this actually all works, because it's pretty amazing. So. All right, so having said that, I'm going to go ahead and get started. I want to be sensitive to people's time to get out of here and go where you're going afterwards. So there we go. Okay, so here's where we left off. We were talking about the Ttam Framework, which stands for tracking, targeting, assembly, and modification, which, as I described, is a way to sort of collect all these observations about the way organisms are adapting. Now, a lot of this, as I'm sure you noted, is, I think speculative is the wrong word for it, but it's certainly not fully formed. There's a lot of research that has to happen here to understand exactly what's going on. And that's why we've been talking about a framework which allows us to start putting those observations into a sort of a pattern to try to understand what's actually going on beneath the surface. So where we left off was at, there we go, backwards, at targeting. We were still talking about targeting. We had talked about tracking, now we're on targeting, then we're going to talk about assembly and modification. Where we left off on targeting was we were talking about predictable, repeatable, reversible solution sets, and now I want to mention just a couple of examples of precise control that's happening. So, as an example, we're going to go right back to the beak shape in songbirds, not just Darwin's finches, but all songbirds. If you remember from the quote that I had out of the study, it's not just Darwin's finches, but all songbirds. So remember that with those songbirds, there we go, that all of the beak shapes could be mapped onto a parabola. They're a conic section. It's very precise in shape as well as in mathematical function. If I can get the mathematical function up. There we go. So remember, it's controlled by a function. So this can be described by math. It's precise. Math is very precise. It's precisely controlled. All of those beak shapes fit this function, and the organisms are generating those during development. So it's precisely controlled. There's another example which I had talked about last time I was here. There we go. 5 years ago, and that is the relationship between the cephalofoil and the pectoral fins in hammerhead sharks. The cephalofoil is the big hammer of the hammerhead, right? So let me just pop up a couple slides from my last talk, if I can. There we go. So those are some hammerhead sharks. There's a lot of variation in hammerhead sharks, specifically with the cephalofoil. So here you've got your great hammerhead, your winghead, whose cephalofoil is almost as long as his body. And then on the other hand, you've got the scoop head and the bonnet head, especially the bonnet head, which you can barely see any kind of extension from the head at all. So there's a lot of variation in these sharks. But what's also interesting about these sharks, there we go, on the left-hand side is the variation from the bonnet head all the way down to the wing head. You can see the differences in shape, and this is the actual measurement between 19.3, I believe that's inches, and 44.4 inches. That's a pretty big spread. But what's interesting is if you take the cephalofoil area, the area of this, and the area of the pectoral fins, it's always a constant. So it doesn't matter how big the cephalofoil is, the pectoral fins do the opposite, so the surface area is always the same in terms of a constant. So the large winghead shark has small pectoral fins. The small bonnet head has large pectoral fins. And it strongly suggests that the variations going on here is controlled by a pre-programmed function or algorithm. So that's another, there are a lot more examples like that, but I want to actually get into what's causing this as opposed to continuing to describe it, right? So let's move on to the assembly part of the framework. And there I said that adaptation occurs during development, not reproduction. And that's significant because it's exactly opposite of what you've probably been taught. There we go. So most variation occurs during embryological and juvenile development. And we just gave a bunch of examples of targeted response of that exact thing happening. Remember the fish, it was in the egg. The beetles was during larval development. The fish, the Omicus was during juvenile development. It all is all happening during embryological and juvenile development. It's not happening during later in life or during reproduction. So what we're discovering is that organisms, they respond differently to the different environments, and they respond the same environments or similarly. So it's the response that determines the traits that are expressed. This happens during development as these traits are expressed and developed as they go on. It has nothing to do with the environment per se. It has everything to do with the organism's response. So what I'm saying is that these traits are expressed during development. They're not selected after the fact. There's no selection going on here. The organisms are controlling the expression of the traits. So this is happening as the traits are being assembled by the organism. Okay, and then lastly, modification. And here I said adaptation is adjustable, not static. So the idea that you've probably heard is that the creatures have this variation. They go out there, they're stuck with what they've inherited from their parents, and natural selection does its job and weeds out those that aren't fit. What I'm saying is that is not true. Those adaptations are not static. So there's lots of examples of organisms that experience change in their environment and they self-adjust, and then they pass those adjustments on to their offspring. There's an entire field of study about this. It's called epigenetics. And again, because we're going to talk about the mechanism here, I'm not going to go a lot into epigenetics. I'll maybe give you one example. But it's very well documented. There's a lot of research going on in epigenetics. And essentially, it's sort of like priming the pump for the next generation is what's going on with organisms. So, for example, you could take a guinea pig, which, not ours pets, they actually live out in the wild as well. And you can put a guinea pig in a situation where you warm up the habitat they're in. The guinea pig adjusts to that warming, and the way it adjusts is by suppressing the expression of some genes and enhancing the expression of others, and then it passes that adjustment on to its offspring. So it's modifying its own genetic code and passing that modified code on to its offspring. But I want to give you some examples of organisms doing the same thing in real time, modifying their own traits in a very dramatic way. So you may not think this is dramatic, but I do. The snowshoe hare is used often as an example of natural selection. And the reason why it's used that way is because we go back again to natural selection's favorite example of predation, right? So a snowshoe hare lives where it's snowy. And so they're white so that they don't get eaten, right? But what most people don't realize is snowshoe hares don't live in the Arctic Circle where it's white all the time. They live on the edges, and so it's not always white. So in fact, snowshoe hares have two variations, not of different kinds of snowshoe hares, but in the same hare. So because their survival largely depends on camouflage, in the winter they're white, and in the summer the same rabbit, same snowshoe hare, is not white, it's brown. So here's an example of a snowshoe hare in the winter with its white coat. Here's an example of a snowshoe hare with its brown coat in the summer. So obviously, it has to go from white to brown when the seasons change, right? So I don't know if you've ever seen a picture of one doing that, but here you go. So this is a snowshoe hare losing its winter coat and going to its summer one. Now obviously, the timing of this is important, right? Because right now, that snowshoe hare is not camouflaged in the snow or out of the snow, right? So it's in a vulnerable position. So it's going to want to make this change quickly, right? And that just makes sense. So it's interesting to find out how this change happens. There we go. So the initiation of that change is pretty much fixed. It's dependent on photoperiod. So basically, it has to do with the change in the season. It's more or less fixed. The seasons change the same time every year because it's the sun, right? The sun isn't affected. However, the speed of the change, the rate of the change, is not related to photoperiod, but temperature and snow cover, which makes sense, right? So what that means is that a colder spring with more snow on the ground, the change happens more slowly. So basically, the snowshoe hares are modifying their color in response to the environment that they see around them. Cold, snowy spring, slower change. Warm, not snowy spring, faster change. But it's even more dramatic than that. This is one of the most dramatic examples you've got, is a sex change. So for instance, a lot of reptiles, like turtles and alligators, their sex is determined by the temperature of their nest. So here's an example of a turtle, and this is the ratio of male. So as the temperature goes up, a peak, more and more males are produced, and then after that it's more females again. Same thing happens with this turtle, only this one it's sort of one way. As the temperature goes up, the ratio of males goes down. Alligators have a spike as well. That's pretty dramatic. Oops. Now it's going from not moving at all to moving really fast. So also, as an example, are wrasse and clownfish, and they change their sex based on the population dynamics, not on temperature in the nest. So again, this happens during development. The wrasse, they are all born female, and they're in a group of fish, a school of fish, and the dominant female changes its sex to male. The clownfish does the exact opposite. There you go. You'll see Nemo in a whole new light. They're all born male, and the dominant one becomes female. And to continue my movie references, what about reed frogs? You remember these from Jurassic Park, right? So in Jurassic Park, the idea was, it's fiction of course, but the idea was that they had missing sequences in dino DNA, so they filled them in with amphibian DNA from the reed frog. And then do you remember what happened? They run around in Jurassic Park and they come upon a nest. Because remember, the dinosaurs supposedly were all female because they withheld a certain, again, they adapted during development, they withheld a certain protein from them, and they were all female as a result. Well, what happened? In an all-female population, at least one of them became male because we had a nest that had fertilized eggs and dinosaurs hatched and left it. So, reed frogs do that. Okay, so, and again, this is now independent of me, just so you know. The slides are changing by themselves. So I was going to give you a nice little introduction to this, but I don't get to. So instead, I will just say that on the right-hand side here, you've got a drawing of essentially the face with the jaw of a fish. I don't know exactly how to say it. I think it's cichlid, but I'm not sure. But anyway, this is I've given you a bunch of examples of organisms that act in response to their environment. They make a change to their genes, their traits, et cetera, in response to their environment. This is an example of an organism which directly manipulates its adaptation. Why do I say that? Well, they've got the jaw right here. You see this is the RA. This is the one that changes the most. It's part of their jaw. And here's what happens. The larvae exhibit a gaping behavior. They open and close their mouths, okay? And this behavior starts before there's bone there, just cartilage. So that as larvae, they come out of the eggs, they're larvae. They have cartilage here, but not bone yet. The bone starts to deposit. During this process, they're opening and closing their mouths. They're gaping. And what's been discovered is, pretty amazing, that gaping frequency varies depending on the species and depending on the individual inside the species. And there's a direct correlation between the gaping frequency and the differences when how the bone is deposited. In other words, jaw shape. So, and what's important about that is that the jaw shape is directly related to their feeding specialization. So they've adapted to the food they want to eat by actually directly manipulating the shape of their jaw with their behavior. So they are actually directly controlling their adaptive traits. Pretty amazing. So here's what the study authors had to say about that. I think. There we go. Okay, they said, although the field of evolutionary biology, again, they're still talking in terms of evolution because they don't know any other terms, even though they can't explain what they're seeing, says, although the field of evolutionary biology is pivoting away from a mostly gene-centric view of adaptive variation, the prevailing paradigm remains focused on identifying genetic sources of such variation. Data presented here, in contrast, show that a seemingly trivial gaping behavior of cichlid larvae can also introduce a large degree of variation. In fact, the magnitude of difference in skeletal morphology induced by these simple shifts in behavior was similar to those predicted to be caused by genetic factors. They're saying the behavior has every much as to do with the shape of the jaw as genes. That's interesting. So, let's put all this together. I've gone through a lot of examples. I've given you a framework. Let's put it together. First of all, the source of the adaptive variation is key. Remember, Darwin didn't understand it, and Darwinism failed. Now, in neo-Darwinism, the modern synthesis, it's been misidentified as random mutation, and neo-Darwinism is in crisis. We just described how it works from a framework, but we still haven't identified what the mechanism is. So let's identify the mechanism. We're going to start by comparing the two different models between non-designed random adaptive process versus what I'm calling engineered optimization. I'm going to say, as I think the examples are starting to show, that adaptive variation is an engineered response. So what do those two different paradigms look like? Well, on a non-designed random adaptive process, you've got passive organisms, and they're shaped by external forces, the environment acting on them. In engineered optimization, you've got active organisms. They're shaped by internal forces. They're responding to the environment. This non-design process is undirected. It's a struggle for survival. It's inefficient. It's by trial and error, hit and miss, and it's slow. It's gradual, successive, slight. On the other hand, with engineered optimization, it's purposeful. They're being optimized to fit an environmental niche. It's targeted. It's predictable, repeatable, reversible, and precisely controlled. And it's rapid. These solutions are fully implemented in as little as one generation. So the framework, to remind you again, also describes what's going on here. We've got tracking, targeting, assembly, and modification. And as I said, the adaptation is responsive, not passive. It's targeted, not trial and error. It occurs during development, not reproduction. And it's adjustable, not static. There's something else we need to talk about, though, when we're looking for a mechanism. We're describing what's going on, but there's a couple of key characteristics of organisms that need to be explained if we're going to come up with a mechanism that describes what's happening here. So first of all, we have adaptive change, which we've just been talking about, and we've seen that it's rapid and it's targeted. It's targeted variation. But also there's divergence. So speciation happens. So if organisms are in control and doing all their adaptations and engineering optimization, how do we explain how they diverge? How do we explain speciation? How do we explain those mashups, for instance? In addition to that, though, with all this change going on, we also have to keep in mind there's something called stasis, which is a cat is a cat is a cat, right? As it adapts, same thing with dogs, et cetera. And also that adaptation isn't necessarily always going on all the time. Sometimes populations are stable and don't adapt for quite some time. So how do you explain that? That's stasis, but also allowing rapid and dramatic change to the point where sometimes they diverge. So what we need is a mechanism that explains all three of those. So is there one? Yes, it's Bitcoin. Now, I'm only partially kidding. So why do I say Bitcoin? It's really interesting that this is in fact the solution. It's not really Bitcoin itself, but what's behind Bitcoin that explains what's going on here. So what's behind Bitcoin? It's run by something called blockchain, which is a distributed problem-solving algorithm. It's computer code, distributed, and problem-solving. And blockchain can be used not just in Bitcoin and other digital currencies, but anywhere where you need what's called a distributed consensus. We'll come back to that in a second. And that is actually what stops hackers from creating Bitcoin for themselves and making themselves rich, is that the number of computers involved, number of computers that the algorithm is distributed to is so large, and they all come up with the same answer, that trying to come up with a different answer, you would need to overwhelm that very large computer network. It's next to impossible to do. So how does blockchain work? Well, I've hinted at a little bit. It's on a network of computers, which they're called nodes in blockchain, and they maintain a single record of transactions. Again, it's primarily for accounting purposes, money purposes, tracking, in other cases, tracking, a chain of supply, a supply chain, things like that. But they keep something called a ledger, which is basically just exactly what it sounds. It's a ledger. It's a record of transactions. And they're maintained on every node. In addition to that, they group these transactions into something called a block. And that block is then chained to the previous block, hence the name blockchain. So what does that look like exactly? So you make a new block, which is a group of transactions, remember, when a complex mathematical problem is solved. And I don't know who here is familiar with Bitcoin, but they have something called mining. They put all these computers, they put all this power, and they're actually even, I work for, National Lab, and we're even talking about small modular reactors. There's tiny little reactors that BitChain folks are thinking about powering their operations with small nuclear reactors. It requires that much power. So it's a complex mathematical problem they have to solve to make more blocks. So how does that work? They mine it using data from the previous block and an irreversible mathematical function. It's a cryptographic hash function, is what it's called. So that means that each block has information from the previous block, and it's connected to that block in an irreversible manner because of the way the math works. So even though Bitcoin isn't actually the answer to how adaptation works, it's actually a close analogy for it, because what appears to be going on is actually a distributed algorithm that allows organisms to generate these adaptive variations. So because of that, I'm going to call it a trait chain, not a blockchain, because we're not making blocks, we're making traits. So I'm going to call it a trait chain. So what is a trait chain? How does it work? So let's compare the two. Blockchain on the left, trait chain on the right. So a blockchain uses a network, which is a group of connected computers. By analogy, in a trait chain, it's a population, which is a group of connected organisms. Okay? A blockchain uses a node, which is a discrete computer that generates blocks. In a trait chain, it's an individual organism that generates traits. A blockchain uses a ledger, which is a digital file that records transactions. Organisms use DNA, which is a molecular structure that records variables. A block is a group of transactions linked to a previous block. A trait is a group of variables linked to a parental trait. Mining is a process of solving those computational problems to get more blocks, and basically what the computers do is they essentially guess a number, but they have to do a lot of guesses to get it right. In trait chain, development is a similar process because the organisms are solving a biological problem in order to generate a trait that's appropriate as a solution, and they arrive at that by adjusting their internal state. I'll come back to that in just a minute. So in blockchain, it's a cryptographic hash function, which is a mathematical operation that's irreversible. It results in an alphanumeric code. In the case of organisms, it's an adaptive hash function because it's irreversible biological operation, and it results in a unique and optimized DNA code. So for those math geeks, we're going to get into exactly what that math looks like, okay? I'll take it easy on you. So if the blockchain is a process that is analogous to the way in which adaptation occurs, a specifically engineered variation, then the following is true. Adaptive traits would result from a biological function based on these inputs: inherited information from the parent, the previous trait, the second one's a mystery. There we go. Information gleaned from sensing the environmental condition. Those are the new variables. And the internal resources available to the organism at the time the trait is generated, which is the internal state. So I promise you math, here it is. So this is the blockchain function. This is what runs Bitcoin and Ethereum and real estate transactions and all kinds of stuff. So it's a function. So there's a threshold on the left, which is greater than the answer to this function on the right. The function consists of a previous block and the new transactions and a novel value, a unique value, okay? In the trait chain, by analogy, you've got a similar function. There you go. It has a constraint on the left. It has a function on the right, and it has variables. Those variables correspond in the following way. The previous block is the previous trait, the trait inherited from the parent, the information for that. New transactions are the new variables, which the creature has used its sensors in the environment. Again, when I talked about it, triggered a cascade that happens when stimuli meet a certain threshold. That cascade that happens, a biochemical response, is the new variables. And when they don't detect an input, they don't change. But if they do detect an input, they then have to take into account their internal state at the time, the resources they have available, their internal programming, their molecular code, the genetics, the variables, all that stuff that's internal to them goes into this equation. Okay, so here's the equation. A constraint is greater than the function that has to be solved of a previous trait, new variables, and internal state. So the tracking that I talked about earlier in the Ttam framework occurs here, the new variables. They're tracking the environment, biochemical response cascade, that's generating the new variables as part of this function. The targeting part is a combination of those new variables and the previous trait. The organism, with those two things in mind, is its internal programming is giving it the solution set appropriate to what it just sensed. And that solution set is partially or even primarily dependent on the previous trait that they had. They don't suddenly become something else. They're modifying, adapting what they already were, that they inherited that information from their parents. And then the actual assembly part I'm talking about occurs when those solution sets from tracking and targeting are combined with the internal state of the organism, and that is the input to the equation. But the question becomes, how do organisms solve that function? Remember, in Bitcoin, you got computers that are guessing these random numbers. How do organisms solve the function? Well, the answer takes us back to what I told you to hold on to way at the beginning, and that is adaptive mutation. Now remember, mutation just means change. Okay? It doesn't mean it's random. It just means it's a change in genetic information. So let me give you a couple of quotes of evolutionary biologists that are seeing this happen but don't get what's going on. Here's how they describe it. It says, the impact of transcription on DNA damage is well understood, but our research reveals a pathway by which mutations can be directed to particular loci in particular environments, and furthermore, that this mutagenic process can be regulated through histone acetylation. Stimulated CNV therefore represents an unanticipated and remarkably controllable pathway facilitating organismal adaptation to new environments. What's he saying? He's saying mutations are happening in the organism, change to the genetic code, in direct response, in controlled response, on specific pathways in response to what's happening with the environment. Evidence for adaptation through genome-wide non-random mutation is substantial, particularly in bacteria. So again, adaptation, the organisms are solving the trait chain function by rewriting their genetic code. Non-random, genome-wide mutation or change in their genetic code. The organisms are rewiring their genetic code. He goes on to say, mobile DNA in the genome is subject to RNA-targeted epigenetic control. Remember epigenetics? How they basically prime the pump for the next generation or even change their own traits to adapt to the environment they're in. So epigenetics. This control regulates the activity of transposons, retrotransposons, and genomic proviruses. Many different life history experiences alter the activities of mobile DNA, and the expression of genetic loci are regulated by nearby insertions. The same experiences induce alterations in epigenetic formatting and lead to transgenerational, passed on to the next generation, modifications of genome expression and stability. These observations lead to the hypothesis that epigenetic formatting directed by non-coding RNA provides a molecular interface between life history events and genome alteration. Again, a fancy way of saying that non-coding DNA is involved in the fact that a creature senses what's going on in its environment, has that function going on, and rewires its DNA to solve the function and create the adaptive variation. Here's another one. Recent applications of high-resolution technologies to genome expression in animals reveal a dynamic, four-dimensional, interactive control architecture incompatible with prior notions that genomes contain discrete functional segments of DNA called genes. What they're saying is that what you've been taught about genes controlling genetic expression and traits is wrong. The entire DNA is involved. We used to have been taught that most of DNA was junk and random, and that there was, these little islands of genes in between. What we're actually discovering is the entire DNA is actually not junk. It's higher level instruction code, and the genes are not that important. They're just variables. That's what we're discovering. So, This sounds an awful lot like a model of DNA that a person named Michael Soltis proposed back in 2014, and he said DNA is really like a computer program. What does he mean by that? Well, in his model, both the instructions and the data are combined into a single stream, just like computer code. What I just said, the entire sequence of DNA, the entire genome is involved in programming the traits for the organisms. So the important part is that though the code is bounded by fixed top-level instructions, input data called variables enable variations. So I don't know if there's any code writers out there, but this is how computer programs work. You have top-level codes that control what happens, but the computer code itself gets variables to do its task and generate its outputs. So here's how he describes it. We have a constraint, and those variables, the formula you just saw, in this case, constraint equals top-level instructions, what he's talking about. So this is constraint, a constraint function, and the variables he's talking about are the previous trait, new variables, and internal state. So here's what he says about his model. He says, using abstraction, computer programs can also dynamically load, move, and control portions of instruction code called subroutines or functions during execution to perform their job. It is even possible for computer programs to generate sections of programming code on the fly, turn them on and off, and call them in different orders, but it is always a computational result of information at a higher level. No matter how many layers of abstraction you have in a computer system, there is always a top-level instruction set that controls everything below it. Why is that important? Because it explains both variation and stasis. So a blockchain-like algorithm, so basically a problem-solving algorithm, what I'm calling trait chain, it's a function in biological organisms, allows for both stasis and rapid targeted variation, which is adaptive change. So when we're looking at these three characteristics we have to explain, we just explained, using a blockchain-like algorithm, both adaptive change and stasis. There we go. But we haven't explained divergence yet. So what is causing divergence? Remember these hybrids, mashups? It looks kind of like two solution sets sort of stuck together, non-overlapping. It ends up looking a little weird, like you slam two things together that don't really belong together. Well, the answer to that lies in the purpose of adaptation. So I'm going to talk about that real quick. So remember our hypotheses back at the beginning? The first one was that adaptive variation is an engineered response to environmental change. And I think that we've pretty much demonstrated that. The second hypothesis was that adaptation is not for the purpose of survival, but for optimization. And that survival is just a side benefit. Of course, if you're optimized for your environment, you're going to survive better, right? Because if something in the environment changes, and you're optimized for the environment, you're going to do a much better job than someone who's not optimized for that environment. You're going to survive better. So this is an example of what's called an iterative analysis. So basically what happens here, I took this from a course about how to become a business analyst. This is applicable to a lot of different things. But you see this is the cycle that happens where you have design, development, testing on an iteration. And then based on what happens with testing, you then go into another design phase, development, and more testing. And you keep doing this iteratively. And as you do so, you get closer and closer to the best solution. Well, as it turns out, that's exactly what organisms are doing. Think about it. They track, they target, assemble, and modify. Then the same individual or the next generation, depending on what the change is and what the modification is, does exactly the same thing. They track what's going on in the environment, which apparently is nothing. I love it how it always shows up right to steal my thunder, right as I'm about to make a point. It's interesting. So they track what's going on in the environment, they target, assemble, and modify again. And this keeps going on throughout their life and the lives of their offspring. in an iterative process all the way through. They keep tracking and doing this. So you may have heard this statement. Individuals don't evolve, populations evolve. Actually, what they really mean is individuals don't adapt, populations adapt. But you know what? They're dead wrong. In the trait change process, it's an iterative process in which the individual organisms are iterating and adapting. So what about populations? If I just said that individuals adapt, does that mean populations don't? Well, no, populations adapt as well. How does that work? Well, let's go back to our blockchain model, because it's a good analogy. It uses something called distributed computing, which uses multiple network computers to solve a complex problem that can't be solved within a reasonable amount of time by just using one computer. So if you've got a really hard problem to solve, you need a fast answer to it. You use distributed computing. You take the same algorithm, you give it to a whole bunch of different computers, they all work on the same problem, come up with their answer, and come back together, right? And that's what enables the execution of what's called a distributed algorithm. They're really good at solving problems where a consensus must be reached. Now remember, blockchain is also a distributed problem-solving algorithm with a purpose of establishing a consensus. And that's established by communicating between those computers, between those nodes, right? That's how they communicate and establish their consensus. Well, does something similar happen with organisms? Absolutely. Trait chain is a kind of distributed algorithm, and organisms establish consensus by reproduction. So what happens is each organism is out there in the environment, it encounters an environmental problem, it generates iteratively its solution set to that problem. We just saw a whole bunch of solution sets in the early examples. It generates that, and because the nuances of the exact problem it encounters are unique, and because its own internal state is unique, the solution set it generates is also unique. However, a similar problem, because this is predictable, repeatable, reversible, and precisely controlled, is going to generate a similar solution set. And all of the organisms generate their similar solution sets. And what happens then is the most common solution sets are reproduced in the greater number. And that allows, in each generation, another iteration of the same thing happening so that the entire population rapidly converges on the optimum solution set to the environmental problem they're facing. So what I'm saying is a population is just like a very powerful distributed computing network solving an environmental problem. And that's why they can do it so fast. And that is, in fact, the purpose of adaptation. They are designed to optimize themselves to the environments they encounter. So that does still leave a question: what happens if you isolate some of the computers from that network? Remember, when we isolate individual organisms from each other, they tend to form species. So what happens when you do that analogy in a blockchain network? Well, the blockchain splits. So in other words, the parameters, the function's the same, the rules about what language to use is the same, it's all the same, but the transactions are different over time. And this happens, you can see this in blockchain, it happens all the time. So that leads to a different ledger. And that may very well be what's going on with species. They have a different ledger, they've experienced different life history, therefore they have different solution sets. And therefore they look different than that group of the population that didn't experience those same environmental problems. In addition to that, it probably very well explains the mashups that we just talked about. They have two separate solution sets, and when you try to put them back together, they have two different life histories at this point. So they're not going to easily merge. They've solved two different problems. So the solution to two different problems is involved when you put them together, and that's why they don't really, they look weird, because they're not optimized to a specific problem. You're trying to put two or more different solutions together in the same thing. But what that also means is we have a great analogy for why you can't turn a cat into a dog. Because In the case of Bitcoin and other blockchain-run digital currencies, there are different functions with different types of rules for making blocks and constraints, et cetera, that run those different currencies. You can't turn Bitcoin into Ethereum, for instance. Even though they use exactly the same kind of function, even though they use exactly the same kind of math, the parameters are different. Therefore, they are different entirely. They're not the same thing. And so they're incompatible. Even though they use exactly the same kind of function, exactly the same kind of rules, they're still incompatible. Which means if you have the same type of DNA setup, but the actual chromosomes and things are different because you have different parameters, even using the same kind of function, that you cannot crossbreed or descend from a common ancestor because they're not compatible. So that means that a blockchain-like algorithm, which I'm calling trait chain, allows for chain splitting, which leads to reduced compatibility, and it also allows for separate blockchains which are entirely incompatible, the kinds, the biblical kinds. So we've talked about adaptive change, divergence, and stasis, and I think I've demonstrated that a blockchain-like algorithm, trait chain, can explain all three of those. So, again, we had two hypotheses. One, adaptive variation is an engineered response to environmental change, and we said yes, we've demonstrated that. Two, adaptation is not for the purpose of survival, but for optimization. Survival is a side benefit, and we have now demonstrated that as well. So in conclusion, here's the answer to the question, what is the mechanism of adaptation? The mechanism of adaptation is engineered optimization as described by the Ttam Framework and the trait chain. Engineered optimization is characterized by being purposeful, highly complex, mathematically precise, and very efficient. So that quote I gave you about the computer model of the DNA earlier, I left something very important out. Let me put it back in, because what I just said leads to an inescapable conclusion. Here's the actual quote. Using abstraction, computer programs can also dynamically load, move, and control portions of instruction code, calling subroutines or functions during execution to perform their job. It's even possible for computer programs to generate sections of programming code on the fly, turn them on and off, call them different orders, but it's always a computation result of information at a higher level created by an intelligent being or beings. Computer codes don't happen by chance. They're designed. No matter how many layers of abstraction you have in a computer system, there is always a top-level instruction set that controls everything below it and must be the result of a creative mind. So what I just presented to you is an extremely powerful argument for design that points to the master designer. Adaptation does not happen by chance. It does not happen by natural selection. It is designed by a creator to allow creatures to optimize to their environments and, after the fall, as a side benefit, survive. So I'm sure, hopefully I blew some minds, because I'm sure you've never heard this before, or at least not in this detail. So I bet there are plenty of questions. So we have time for questions, and I don't know how much time, but I think we have some mics to run around.
Yes, we have two microphones.
So yeah, if you'd like to ask me a question, go ahead and raise your hand up. We'll get a mic to you, and I'll do my best to answer them.
Can we have the yellow mic?